Hey, PBio. Today we're going to talk about viruses. This is your 17.1, 17.2 reading. So to start off, right, we'd say a virus consists of a nucleic acid surrounded by a protein coat. Yep, that's the general term. Let's also remember that they are very small. They're much smaller than bacterial cells, and that's important because they infect bacterial cells. And so viral genomes, they can be made of RNA or DNA, and they can be double or single-stranded. So even if they are DNA, they can be single-stranded. So there's a couple different types, and we often label them based on their capsid. So the capsid is like the covering. And it's often made of protein. So in this first one, the tobacco mosaic virus, we say that it's a helical. It is a helical capsid. And that's because you see its proteins are arranged in this like helical shape. Then we have an icosahedral capsule. Sorry, not capsule, capsid. And that's because its capsid has this triangular structure to it. And an example of this are these adenoviruses. Then we have our influenza viruses, which we'll talk about a little bit later. But you can see that they have an envelope with some glycoproteins. So that's these little guys sticking out. And we will talk about those guys. And then we have our bacteriophages. I'll give you one guess as to what they infect. We often just call them phages. They infect bacteria, and they look kind of sci-fi to me. They have a head with the genome inside of it, and this tail, and these little protein sort of legs that allow them to inject their viral genome into the bacterial cells. And so when we talk about what type of cells viruses can infect, no, they cannot infect any cell. They are specific to what type of cells they infect, and that's because of the proteins on their surface proteins. So we talk about this all the time, right? Structure affects function. We see this firsthand right here. So the surface proteins can only fit into certain receptors. And so this limits what type of cell, like what type of species viruses can infect and what types of cells within those species. So for example, can I infect nerve cells or respiratory cells within the human species? So it limits them to what they can infect. And so when we talk about how viruses replicate, we have to remember that they must have a host cell. And this is kind of like sci-fi almost, how it just sort of takes over its host cell's machinery. So when we talk about protein synthesis, the virus is going to take over your ribosome. It's going to use your tRNA. It's going to use a bacterial cell's nucleotides, whatever it is. And so what it basically does is the virus injects its genome, and the genome is used to make more copies of the virus's genome. And it is transcribed into mRNA, which is going to be translated by the host cell's ribosomes into more viral proteins. So we got the host cell's machinery making more copies of the genome, more copies of the proteins, and then we're going to see the self-assembly. The viruses will kind of get into shape, and then they will, many copies will leave the host cell. And then those viruses can go and infect other cells and do the same thing and trigger infection. Yeah. The host cell, it can be killed, so it can result in death for the host cell or just damage of it. The first replicative cycles that we're going to focus on are of phages, and phages are the viruses that infect bacteria. And these are I want to say organisms because we're on a debate if viruses are living. These are the guys that infect, or that Hershey and Chase used for their experiment to try and figure out where was protein the genetic material or was DNA the heritable material. So keep that in mind. And so bacterial viruses, bacteriophages, have 2 cycles, depending on what they do. So if they kill the cell, then it's a lytic cycle. But if it does not kill the cell, it's a lysogenic cycle. So we'll start with the lytic cycle in terms of what it results in. So the lytic cycle, if you're a phage going through the lytic cycle, you're known as a virulent phage. So it does the lytic cycle only. This T4 guy is an example of that. So what it does is the phage injects its DNA into the host cell, and the host cell's DNA is broken down. Then the viral genome is going to be used to make more protein and more of its own genome. So more viral proteins and more viral genome. And again, this is using the host cell's machinery. So it's ribosomes, it's enzymes, it's tRNA, it's nucleotides. We'll see some self-assembly of the viruses into their final structure, and then the viruses burst out. And so this results in the host cell dies. Well, maybe it's a sad face depending on the type of bacteria. But the host cell dies, and the viruses can then go and infect other cells. If you're going through both the lytic and the lysogenic stage, lytic and lysogenic cycle, you are a temperate phage. So you do both the lytic and the lysogenic cycle. We'll talk about that in a second. So the lysogenic will be over on this side, and the lytic happens over here. So the first thing that happens is this lambda-phage, it injects its DNA into the host. The viral DNA kind of circles up, and then it can be incorporated into the bacterial's actual DNA. So if that happens, so if it proceeds from here to here, meaning it becomes a prophage, which is viral DNA or viral genome and the bacterial genome and a circular chromosome, then we say it's in the lysogenic cycle. And so the lysogenic cycle, what's going to happen after we get that prophage is the prophage or the bacterial cell will replicate like normally. And the result is many copies of this prophage. And that prophage, again, is just a bacterial cell with the bacterial's DNA and the viral DNA. And so during this time, the bacterial cell isn't making viral proteins. But if there's some sort of triggers, maybe there's an environmental chemical or some sort of radiation, the bacterial cell or the prophage can enter the lytic cycle. And that's why we call it temperate. It's not in the same at all times. And so it's triggered by something in the environment usually. And so it goes towards there. And then what we see many copies of the virus being made until the viruses burst out and the host cell dies. And then those phages can now go and infect other cells. So that's a temperate phage. Animal viruses differ from bacteriophages in a couple ways. The first way is that animal virus genomes are more often RNA. And animal viruses have an additional envelope. This envelope means that it has a membrane coated with glycoproteins. And those glycoproteins, like all your cells have glycoproteins that say, hi, I am a skin cell that belongs to a SIPL. But these glycoproteins help give a cell its identity, right? And so as a result, whatever these glycoproteins are on the virus, this affects what type of cells viruses can infect. So the general thing that the animal virus does is it infects the host cell. It sheds its envelope, usually sometimes merging with the host cell's coating, and the viral genome is now in there. And so the virus comes with something called RNA polymerase. RNA polymerase is different than your RNA polymerase. So this is viral RNA polymerase. It can use RNA as a template to make RNA. Your RNA polymerase uses DNA as a template to make RNA. So if we answer this question, this virus uses a viral RNA polymerase, we would say it can use RNA as a template to make more RNA. So once that viral genome is injected into the cell will make mRNA, and then it will translate it into glycoproteins and capsid proteins. So it will use RNA polymerase to make mRNA, and then it will translate those mRNA molecules into capsid proteins and the glycoproteins that the cell the virus needs on the outside of its envelope. The other thing that it will do is it will make another copy of the RNA genome so that it can make a lot of copies of the RNA genome that can be used in the production of the new virus. So then what happens is, as this all is happening, we see self-assembly over here. The capsid coats the RNA, and then the glycoproteins are attached to the host cell's membrane, which then, as the new virus leaves, it actually has part of the host cell's membrane with the glycoproteins on the outside so it can bind to other cells. Inside of the virus, we see its capsid made of proteins and its viral genome. This is not like the lytic cycle, aka the one that kills the bacterial cells, because it doesn't kill the cell usually. So this is one of those RNA viruses. Another type, herpes virus. So it is actually a type of virus that leaves mini chromosomes of viral DNA. So herpes viruses leave their viral DNA inside the nucleus of host cells. If they are triggered, and so a trigger might be, the host cell is under a lot of stress or something in the environment, some sort of chemical. If it's triggered, it will make more copies of the virus. So if it makes more herpes virus copies, then those herpes virus copies can leave the cell, and then it can go and infect other cells in the host, maybe causing something like a cold sore or another phenotypic effect. Going back to RNA genomes, There's 3 different types. So one is that the RNA genome can be, it's mRNA already. And so as a result, it can be translated into viral proteins immediately upon injection. The second one is RNA that must be transcribed through transcription into mRNA, and then it can be translated. This is different than our central dogma because it is something where RNA is used as RNA, and this requires RNA polymerase. And again, viral RNA polymerase, different than yours. The third kind is, these guys are called retroviruses. And no, not from the 70s. Retroviruses, they use reverse transcriptase, which is an enzyme that can take RNA and make DNA. So what happens in retroviruses is they use reverse transcriptase, which is, you guessed it, an enzyme. This reverse transcriptase comes with the virus, and basically what the reverse transcriptase does is, using the machinery, so like the nucleotides, the ATP, all that good stuff from the host cell, it takes its RNA template and makes it into DNA. We will see this with HIV, so we'll look at HIV in a second. HIV is a retrovirus. HIV is called, it's short for human immunodeficiency virus, and that's because it infects white blood cells, which are a vital part of the human immune system. They are your defense. Because HIV infects white blood cells, it causes the disease AIDS, which stands for acquired immunodeficiency syndrome. So it's acquired because it's a virus that you can get, and it causes deficiencies in your immune system. It makes it harder for you to fight off infection, and so that's where it gets its name. So what does HIV, the virus, do? Well, basically what we see is just like what we were looking at before, it injects its RNA into the cell. Boom. What happens is reverse transcriptase makes a single strand of DNA from the RNA. That's right here. So after it injects the reverse transcriptase, the enzyme that came with our HIV, we see it right there, it makes DNA from RNA. Then we see reverse transcriptase again is used to make a double-stranded DNA. And once that happens, the DNA, the viral DNA goes into the nucleus and becomes a part of the host cell's chromosome. And so we call it at this point a provirus. So a provirus is the HIV genome plus the host cell's genome in a chromosome. Well, what happens after that is just like what we talk about with transcription and translation. So once the HIV genome is incorporated into the host cell's genome, what we see is the provirus is able to make copies of HIV's DNA and mRNA. And then that mRNA is able to be translated into HIV's proteins. So both its glycoproteins and its capsid proteins. And just as we saw before, the HIV new virus will leave the cell via exocytosis and it will be fully assembled. And now the new virus can go and infect other cells. And that's HIV. The last question's on here. What kind of cells does the common cold infect? So the common cold infects epithelial cells of your respiratory tract. And so this actually gets to an important point about viruses. Depending on what type of cells viruses infect depends on how bad they are. That's part of what depends on how bad they are. So your epithelial cells of your respiratory tract, those are really fast dividing cells, and as a result, you can repair the damage from a virus and you recover from a common cold. Other viruses infect nerve cells, and so as a result, those viruses, those are permanent damage because nerve cells, we know, don't divide very frequently.